Hello, everyone. My name is Sean Kosh, and I'm a second-year graduate student in the Master's in Integrative Biology program at Kennesaw State University. My poster presentation is titled RNA-Seq Transcriptome Analysis of HEK-293T cells after hemorrhagic snake venom injury. This work can be applied to the field of wound regeneration, where we can study the wounding process by using hemorrhagic snake venom to model acute injury. And with the battle against COVID, snake venom can be used to help develop drugs to assist in providing immunity. In the past, snake venom has been used in various studies examining cancer progression. In our study, we examined the molecular signature of human embryonic kidney cells in response to Crotalus atrox venom stimulation. We can see this in the first figure to the left, where the first step was testing the bioactivity of our venom by examining various concentrations outlined here, 5, 10, and 20 micrograms per mil. We saw that 10 micrograms per mil produced sufficient damage to the monolayer without causing mass apoptosis. This was our chosen concentration. And we can see that with the images here, where in 10 micrograms per mil, we did not see excessive tearing of the monolayer like we saw in 20. Transcriptome analysis of Crotalus atrox stimulated and non-stimulated HEK-293T cells showed that there are 243 differentially expressed genes with 137 upregulated and 106 downregulated. And over here, we outlined the 9 most significant upregulated genes in response to our treatment, as well as the 9 most significant downregulated genes. These gene groups included those involved in early growth response, heat resistance, apoptosis, and the regulation of zinc. This can also be seen here according to our gene ontology analysis, which showed that out of the 29 upregulated genes, 24% consisted of proto-oncogenes. We can also see here that out of the 29 downregulated genes examined, 30% were associated with the ATP binding cassette family. HEK pathway analysis identified 147 total pathways with 8 significant ones and 139 not significant pathways. And out of these pathways, we can see here the 8 that are outlined that were significant. The mucin type O glycan biosynthesis pathway was the most significant out of the 8. There are 4 genes identified in this pathway that we can see here. And the only gene that showed an upregulation was GAL-NT4, and it also showed the greatest fold change, 3.08. And this gene is typically associated with nephrosis, which has to do with kidney damage. And overall, our data suggests that there's an upregulation and downregulation of a wide variety of genes, as we have outlined. And the significant movement in the mucin type O glycan biosynthesis pathway tells us that our treatment may have an impact on nephrosis, which is associated with kidney disease.